Now let's discuss about allosteric enzymes. In non-competitive inhibitor, we saw allosteric site means other site other than the active site. So here the same concept we are using, and then we will discuss what do you mean by allosteric enzymes. Before starting this, please see this diagram carefully. Now this red part, it is the enzyme part, and this enzyme part, it has this site where blue color structure is attached, which is called as active site, and at the bottom it has one other site which is called as allosteric site. Okay, so enzymes, this enzyme, it has two sites, that is two receptor sites. Enzyme, allosteric enzymes, these have two receptor sites. Okay, one site where substrate attach, that is called as active site, and which is present in almost all enzymes. But allosteric enzymes, they also possesses some other sites, and to these sites either an inhibitor is attached or activators are attached. Please don't get confused here. This is the active site where substrate molecules are attached, while in allosteric enzyme they have some other sites where either inhibitor or activator molecules are attached. Now when these molecules, when inhibitor molecules are attached to the other site, then the shape of the active site gets changed and substrate molecule cannot attach to the active site and there is no reaction. This we have discussed in non-competitive inhibition. These allosteric enzymes which has other shapes or conformation induced by the binding of modulators, which either of inhibitor type or activator type, and allosteric enzymes are very important in feedback regulation process. Actually, feedback regulation process in detail is not included in our syllabus, but still, what is feedback regulation? If there is a series of reaction, like A get converted into B, B get converted into C, C get converted into D, and D get converted into E, that time the end product, that is E, control the reaction of A, that is the starting compound, and that is called as feedback regulation. Feedback regulation is very important for the regulation of many activity. So what we were discussing here, allosteric enzymes, the enzymes which has other site other than the active site, those are called as allosteric enzyme. Now allosteric enzymes, they are of two types. One is allosteric inhibitor and another is allosteric activator. So first we will see what do you mean by allosteric inhibitor. Now before this, please see this reaction. Here this is the enzyme. Can you see? This enzyme has two sites. One is active site and this is other site. To this other site means this is the allosteric enzyme because it has active site as well as other site. Now to this active site, if inhibitor is attached, so what happened here? There is a formation of enzyme inhibitor complex. But here when there is a formation of enzyme inhibitor complex, it changes the shape of the active site. Here you can see the shape of active site. It is very much suitable to the substrate. But as soon as inhibitor is attached to the other site, it changes the shape of the active site. Now substrate cannot attach to the active site and there is no reaction takes place. So these inhibitors, what they are doing? They are changing the shape of the active site and they stop the reaction. So we can say that allosteric inhibitors, it is a type of non-competitive inhibitor which attaches to the enzyme at allosteric site. Allosteric site means the site other than the active site and inhibit the reaction, that is called as allosteric inhibitor. The best example of allosteric inhibitor, it is observed in glycolysis process. In glycolysis, ATP, it acts as a allosteric inhibitor for the enzyme pyruvate kinase. Now, what is the function of pyruvate kinase in glycolysis? Pyruvate kinase speed up the last step of glycolysis by transferring the phosphate to ADP to form ATP. In a simple word, we can say that pyruvate kinase, it is involved in the last step of the glycolysis process, that is the metabolism of carbohydrate. And here what it does, it converts ADP into ATP, diphosphate into triphosphate. But the ATP which is formed, it itself distort the shape of the active site of pyruvate kinase. Once the ATP is formed, what it does? It attacks the pyruvate kinase enzyme to the other site, that is allosteric site, and it distorts the shape of the active site of the pyruvate kinase and acts as a allosteric inhibitor in this case. So this is the best example of allosteric inhibitor where it decreases the rate of reaction by attacking on the other site of the enzyme, so the shape of the active site gets changed. But there are some other allosteric enzymes which are also called as allosteric activators or allosteric effectors. Now, what do you mean by according to its name? Like inhibitors inhibit the process while activators activate the process. So, allosteric effectors, these certain substances which are also considered as allosteric modulators or allosteric effectors, they bind to the allosteric site of the enzyme and regulate or sometimes increases the enzyme activity. Now please see here. Here two reactions are given. This inhibition process we have already discussed. Here you can see this is the enzyme. When allosteric inhibitor attached to the other site, it changes the shape of the active site and the rate of reaction is decreased, because when allosteric inhibitor is attaching on the other side, active site, its conformation gets changed, which is not suitable for the substrate. But here if you see in other reaction, here again this is the enzyme and here allosteric to the allosteric site, this group is attached. But when this is attached, it is not changing the shape of the active site. Substrate can easily attach. Active site, it has the very correct conformation, and such compounds which regulate the enzyme activity, those are called as allosteric activator. So we can say that allosteric inhibitor inhibit the process, allosteric activator activate the enzyme activity by attaching to the other side. Now, allosteric effectors, there are two types of allosteric effector. When the enzyme activity is increased, If the enzyme activity is increased, then it is called as positive allosteric effectors. And positive allosteric effectors, they increases the rate of reaction. On the other hand, negative allosteric effector bind at the allosteric site or inhibitor site and inhibit the enzyme activity. So we can say that here, when allosteric activator is attached, then this site is called as allosteric activator site, allosteric activator site, and when inhibitor is attached, then it is called as allosteric inhibitor site. Because it is increasing the rate of reaction, so it is called as allosteric activator site, and this is called as allosteric inhibitor site. Allosteric enzymes, they are again of two types. One is called as K class of allosteric enzymes, K class, because these effectors changes the Km and not the Vmax. Double reciprocal plots, their double reciprocal plots are similar to competitive inhibition. If you remember competitive inhibition, because K class allosteric enzymes, they changes the Km but Vmax remains constant, while V class allosteric inhibitor, they changes the Vmax but Km remains constant. That means their double reciprocal plots resemble to that of non-competitive inhibitors. So, allosteric enzymes, they are of two types, K class where Km is changed and V class means where Vmax is changed. When Km is changed, it is just similar to competitive inhibitor, and when Vmax is changed, it is just similar to non-competitive inhibitors. So, here we have discussed about the allosteric enzymes. Thank you so much.